The field of aging research has experienced a remarkable shift over the past decade, and at the center of this transformation is vitamin B3. Far from being a simple nutrient for preventing pellagra, B3 in its modern forms, nicotinamide riboside, NR, nicotinamide mononucleotide, NMN, nicotinamide, and nicotinic acid, has emerged as a powerful modulator of cellular metabolism, DNA repair, and longevity pathways. Breakthroughs in NAD biology, sirtuin activation, and metabolic rejuvenation have positioned B3 as a foundational molecule in longevity science, changing how researchers and clinicians think about aging not as an inevitable decline, but as a modifiable biological process. These discoveries have opened the door to interventions that can enhance cellular resilience, improve tissue function, and potentially extend health span in ways previously thought unattainable. Central to these advances is the understanding of NAD biology. NAD is a coenzyme present in every cell, serving as a critical hub for energy production, DNA repair, and intracellular signaling. With age, NAD levels naturally decline, leading to mitochondrial dysfunction, reduced energy output, impaired DNA repair, and increased cellular stress. This decline underlies many hallmarks of aging, from frailty and cognitive decline to metabolic dysfunction and cardiovascular deterioration. By supplying precursors such as NR and NMN, researchers have found that it is possible to restore NAD pools in tissues, effectively rejuvenating cellular function. Preclinical and early human studies show that NAD restoration improves mitochondrial efficiency, enhances energy metabolism, and reduces markers of oxidative stress, creating a cellular environment more capable of sustaining tissue function and resilience. These findings underscore a profound shift in aging research. Instead of treating individual diseases, scientists can now target fundamental cellular processes that drive systemic aging. Sirtuin activation is another breakthrough linked closely to B3 and NAD biology. Sirtuins are NAD-dependent enzymes that regulate gene expression, coordinate stress responses, and modulate metabolic pathways across multiple tissues. They are integral to longevity pathways, influencing inflammation, DNA repair, circadian rhythm, and mitochondrial function. As NAD levels fall with age, sirtuin activity diminishes, contributing to metabolic slowdown and increased susceptibility to age-related disease. Supplementing with NR or NMN restores NAD, reactivating sirtuins, and enabling cells to maintain energy homeostasis, repair damage more efficiently, and respond effectively to stress. Preclinical studies demonstrate that sirtuin reactivation can improve endurance, enhance cardiac function, preserve cognitive performance, and even slow cellular senescence. These results highlight B3's role not just as a nutrient, but as a molecular lever capable of influencing multiple longevity pathways simultaneously. Metabolic rejuvenation is another area where vitamin B3 has shown transformative potential. Aging is associated with reduced mitochondrial output, insulin resistance, and accumulation of metabolic byproducts that impair tissue function. NR and NMN supplementation boosts NAD availability, which in turn enhances mitochondrial oxidative phosphorylation, promotes efficient energy utilization, and supports metabolic flexibility. In animal studies, these interventions improve exercise capacity, enhance glucose tolerance, and restore youthful metabolic patterns in liver, muscle, and brain tissues. Early human trials mirror these findings, showing improved markers of mitochondrial function, vascular health, and metabolic efficiency. By addressing the root drivers of energy decline, B3-derived NAD precursors offer a systemic approach to metabolic rejuvenation, improving not only cellular energy production but also overall tissue and organ resilience. Vitamin B3 is also reshaping how aging research approaches disease prevention and multi-organ health. Because NAD and sirtuins operate across virtually all tissues, B3 supplementation has potential implications for cardiovascular health, neuroprotection, kidney function, skin integrity, and immune competence. For example, enhancing NAD levels improves endothelial function and arterial flexibility, supports cognitive performance and neuroprotection, enhances wound healing and skin barrier maintenance, and modulates inflammatory responses systemically. This multi-organ impact positions B3 as a unifying intervention in longevity science, offering broad-spectrum benefits rather than isolated disease-specific effects. It exemplifies a paradigm shift in geroscience, where interventions are designed to target the underlying mechanisms of aging, rather than simply treating individual age-related conditions. Another major advancement in the field is the safety and practicality of NAD-boosting interventions. Unlike experimental drugs that may have narrow therapeutic windows or high toxicity, NR and NMN have demonstrated excellent tolerability in human studies, with minimal side effects even over prolonged supplementation. Nicotinamide and nicotinic acid have long histories of safe use, making B3 a feasible and accessible tool for clinical and lifestyle-based longevity strategies. This accessibility allows researchers and clinicians to explore NAD restoration across diverse populations and over longer periods, providing invaluable insights into how systemic metabolic support influences aging trajectories. Finally, ongoing research continues to push the boundaries of B3's potential. Trials are now exploring its synergistic use with exercise, dietary interventions, caloric modulation, and other compounds targeting senescence, autophagy, and mitochondrial biogenesis. Early evidence suggests that NAD restoration may amplify the benefits of these interventions, producing compounded effects on energy metabolism, cognitive resilience, and systemic repair capacity. This integrative approach reflects a broader trend in longevity science, combining molecular-level interventions with lifestyle strategies to optimize health span and functional resilience in aging populations. In conclusion, vitamin B3 has fundamentally altered the landscape of aging research by providing a biologically grounded mechanism to restore NAD, activate sirtuins, and rejuvenate metabolism across tissues. These breakthroughs highlight the possibility of slowing or even partially reversing cellular decline, improving organ function, and enhancing systemic resilience. Far from being a simple nutrient, B3 has become a central tool in longevity science, illustrating how targeted molecular interventions can reshape our understanding of aging and open new avenues for extending health span. As research continues to expand, vitamin B3 stands out as a practical, safe, and scientifically validated cornerstone in the pursuit of healthier, more resilient aging.